Great. So I'll talk about prediction and transcriptomics, which is a little more mature field. At this point, largely RNA-seq. But I want to come back to some of the points that were raised yesterday in terms of thinking about these omics and why we would want to predict rather than exclusively measure them. So in particular with transcriptome levels, we're talking about an endophenotype that really does concentrate a substantial proportion of the non-coding heritability to common disease phenotypes. So in a variety of ways, people have shown that, for example, there's a huge enrichment of GWAS hits that are EQTLs. You can show that the heritability for common traits is substantially accounted for by EQTLs, by SNPs mapping to enhancer and promoter elements. And so the part of the rationale for measuring, but now in addition genetically predicting, is to have more of a sensible way of aggregating genome variation into what then becomes a gene-based test, and for long non-coding RNAs, a unit-based test with an easy-to-interpret direction of effect. So we understand how gene expression levels might be affecting phenotypes like diabetes or asthma over a lifetime. And so this way of aggregating DNA variants into endophenotypes to probe the relationship between genes and long non-coding RNAs and disease puts this more in the flavor of a Mendelian randomization experiment. And it really is a natural way to iterate between human and model systems where our signals often do come at the level of genes, and translating back and forth between human SNP discoveries and model systems has been much more challenging. So just a few simple descriptions of discoveries made in the context of the GTEx project that inform on our ability to genetically predict transcript levels. So the cis expression, that's the regulation of gene expression at the level of local DNA variants, is not a polygenic trait. It is, you're talking more about tens of SNPs that are predictive in the cis space around any gene or long non-coding RNA. So we're not talking about millions of SNPs, we're not talking about hundreds of thousands of SNPs, we're not talking about tens of thousands of SNPs. We're talking about tens of SNPs being the more optimal prediction of the transcript levels at the cis. Now, from the trans perspective, it's harder, but we honestly just haven't had enough power. So there's a group in Europe that has accumulated a lot of array data for peripheral blood and is getting much better power for identifying the trans contribution to gene expression. But this observation means that polygenic models are not the ways to do prediction at the level of transcript levels. Lasso yields the most parsimonious prediction. It's very hard to beat a Lasso model for prediction of transcript levels. The elastic net does essentially as well as a Lasso and allows for more redundancy. That's the model we have generally used, in part because people will come into a study for predicted gene expression with genetics having been generated from different GWAS platforms with different SNPs directly interrogated versus imputed. And our initial studies showed that the elastic net was a little bit more robust than the Lasso to the kind of genetics product that people might use coming into a prediction experiment. Another piece of information that's key in thinking about prediction modeling is that most genes have both local, very local SNP associations to the transcript levels, but also distantly acting SNP predictors. The long-range enhancers really do contribute. So these two statements are both true. Most EQTLs, so the expression quantitative trait loci, are within 50 kb of transcription start and stop sites. And the SNP with the single largest effect on most genes, transcript levels, is sufficiently distant to that gene to be a no LD with any of the variation in or near the gene. So what you have are often a whole set of SNPs that act quite locally on transcript levels, and then one or more distantly acting enhancers, and variation in and near those enhancers can have a big impact on genes expression as well. And those two things contribute differentially to what happens to gene regulation when you consider it across cell types and tissues versus the very tissue-specific or cell type-specific things. So the local SNPs tend to affect a gene's expression across all cell types and tissues in about the same way, whereas the distantly acting enhancers tend to be much more cell type and or tissue type specific. Most of this looks like it's happening at the level of cell types. And of course, tissues are made up of different cell constituents, so we're often seeing average effects over the cell constituents of any particular sample. But with more and more data being generated at the level of the cell, it's possible to deconvolve these things and understand what are the drivers of the sort of averaged overall cell types that we measure in something like GTEx, which starts out as tissue measurements. And so a brief history of how transcriptomes have been genetically predicted. So the polygenic risk scores versus sparser models. Sparser models were definitely shown early on to have much better quality prediction. But the first sparse models were all built within individual tissue types or cell types. And a recent approach from Hongyu Zhao and colleagues at Yale, very nice paper, showed that if you borrow strength across all the tissues in GTEx by assuming that the effect size for a given SNP across tissues is independent. So the beta for the effect size is cell or tissue specific. You actually do get a better prediction than if you build the model separately in each of the tissues. And building on that, we've then used the information that the local SNPs really are acting across all tissues, and therefore you could assume that the local SNPs are acting with a betas equal across tissues model, whereas the distantly acting SNPs tend to be the cell or tissue specific ones, and there you have to have the tissue specific betas. And that model wins out substantially over a simple beta cell, the beta model with cell or tissue specific effects. So this is a thing that will keep going. So the quality of prediction will increase. Everybody's working on getting the best models. But in general, the improvement in prediction quality is greatest for cells, tissues, and genes in which we had the least power to begin with. So whether you're looking at this at the level of cells, where we're still working with relatively modest sample size, or tissues, and for different tissues in GTEx, we have different sample sizes, and so different powers to build anything. Genes with more modest expression within a tissue will tend to have the lowest estimates of heritability because we don't have enough variance to see much heritability. All of those situations is where we gain more power by improving the quality of prediction. But when we already had high heritability to begin with, if heritability was 40, 50, 60%, we're not gaining anything with these models. So high heritability always yields really high quality prediction performance. So one of the things that is relevant, though, is that genes formerly characterized as not having any EQTLs, actually, when you borrow strength across tissues, often have substantial numbers of EQTLs. And that's actually a really important and interesting class of genes that in the first rounds of GTEx we're not showing as having EQTLs at all, because the people were looking at these more tissue specific. And so that's a nice outcome of some of these more recent predictions. So to show some part of the space where we can use transcriptome data to do predictions, this is a study that grew out of collaborations we're developing with the Mendelian sequencing projects in the genomic sequencing project space. So the question from one of our colleagues at Hopkins was about the phenotype VACTERL. So VACTERL is diagnosed in individuals that have at least 3 of a set of congenital defects, vertebral defects, anal atresia, cardiac defects, etc. And so we start out using the phenome risk score concept to develop a phenome risk score for VACTERL. And so you start out using all of the people on whom we have electronic records, but removing those in whom we have genetic data. So we're now using just the synthetic derivative minus BioView in Vanderbilt parlance. When we identify all subjects with the VACTERL diagnosis or with the term VACTERL included in the physician notes, and then a postdoc went through and read all of the physician notes to determine which of the subjects had truly been diagnosed with VACTERL, and the others for whom it might have been a rule-out diagnosis. So identified all of the sort of gold standard VACTERL cases that it was possible to find, and then identified the codes significantly associated with the true VACTERL diagnoses, and that those codes were then used to create a phenome risk score. So significantly associated codes were included. And then we deployed the VACTERL phenome risk score in the genotype subjects in BioView that we then tested using genetically predicted expression of the genes. So the idea was to get a gene-based result to our colleagues who were doing sequencing of VACTERL subjects to see whether the altered expression of genes associated to VACTERL might sometimes come up as genes in which rare variants were being discovered. And so there, in fact, is quite a number of strongly associated SNPs and regions. So you see HLA, the HLA region come up. But some of these genes are actually quite interesting in that a couple of them have already been identified in the literature as being associated with either early pregnancy loss or some congenital anomalies already, recurrent pregnancy loss, which of course is going to be one of the canonical phenotypes associated to anything that is generally going to be associated with multiple congenital anomalies, because a pregnancy loss will be more common in many cases than a live birth. We also saw several, this is just one example, of the Mendelian an already known Mendelian condition that has some congenital anomalies associated with it, being found to be associated to this phenome risk score for VACTERL. But another of the genes that was quite interesting and a kind of a gene by environment is the Toxoplasma gondii receptor, which is kind of a chilling thought for multiple congenital anomalies. So this is, a brand new example of how we're trying to use gene-based phenome-wide associations to collaborate in studies of genes, so rare variant studies. We're also collaborating extensively both with zebrafish and mouse model systems groups, because again, the opportunity to go across the entire medical phenome is what biobanks do really uniquely well. So rare coding variants or function-rich omics going in and phenome coming out. So it's one thing to look at slices of disease, but with the entire medical phenome, we can see people with multiple congenital anomalies. We can put together biochemical abnormalities with congenital anomalies. We can look at the phenome for genes that in my colleague's study on 500,000 islets isolated from diverse outcrosses, all of the really interesting measurements they've made in those islets and all of the cool genes that they have, we can look at the entire medical phenome associated to those genes and see, really interesting patterns. So I think the opportunities for using biobanks and also the large GWAS cohorts with imputed, not just transcriptomes, but metabolomes and protein measurements, I think it's really fantastic. There's a lot to be learned from the Mendelian randomization part of that, as well as the direct measurement. So with that, I'm happy to take questions.
Mark?
So one of the interesting observations about VACTERL is discordance in monozygotic twins. And so I'm curious about how this type of an approach might help to explain that observation.
Yeah, so I think de novo variants would not be a surprising observation for VACTERL, but the reduced expression of some of the genes implicated in, we already know the reduced genetically predicted expression of some genes that have been implicated in other disorders as de novo mutations, seen almost exclusively as de novo mutations, are associated with many of the same features of those conditions when you can look statistically at tens and hundreds of thousands of individuals. So I think the sort of, you have a one-off observation, and there could be many different genes at which a de novo mutation might lead to something that would be diagnosed as VACTERL. And by seeing lots of genes highly significantly associated to this VACTERL phenome risk score, the possibility that you would get a double hit by contrasting it with the BioView observations and having at least a patient seen with a de novo mutation in that gene, I think is an interesting possibility for being able to move forward.
Dave?
Nancy, that was great. I'm just wondering, in the...
Could you please use your microphone?
Yeah, lean in.
In the analysis you showed us for VACTERL, the first thing you did, as I understand it, is sort of develop a training set from individuals who have some mention of that diagnosis in their record. So, of course, I'm sitting here thinking, well, a mention is not the same as a diagnosis.
No, that's right. That's why they read all of the physician notes to make sure that there was actually a diagnosis of VACTERL for the patient. So in some cases, there was a code for VACTERL. I mean, in most cases, there was actually a billing code for VACTERL. But in some cases, the patient was being seen later for the consequences, the downstream medical consequences of some of the congenital anomalies, and they weren't billed for VACTERL, they were billed for whatever it was they were seeing, but in the physician's notes, they said the patient has a diagnosis of VACTERL, and we're following up on, whatever it is.
So it wasn't a random mention in a differential
No.
diagnosis. It was
No, in fact, those were the ones that were taken out
Okay,
and,
all right.
in building it, because there were plenty of situations where VACTERL was considered as a differential and ruled out. That was the point of reading 800
And
records.
what was the N that you ended up with?
About 876
Ah.
cases for building out the phenome risk score.
There's one more question from Naomi.
I was just wondering, does it make sense to you that the trans EQTLs are tissue specific? Is there a missing step?
Oh, no,
How
so,
do you-
sorry. So distantly acting is
Distinct-
still within 1 megabase.
Oh, okay.
So it's still cis, it's just more distantly cis, sufficiently distant cis that it's usually in no LD with any of the other SNPs included in the model. So most of the models include a set of locally acting and one, and sometimes more than one, distantly acting. And you'll often have the same set of locally acting SNPs in all of the tissues. And then 4 or 5 of the tissues in which the gene's expressed might have a 5' distantly acting enhancer, and another 4 or 5 tissues might have a 3'.
Okay.
So that
makes
they're
sense. Yeah.
much, within those sets of 4 or 5,
Okay.
they're much more highly correlated than across the 2 sets because of those distantly acting enhancers.
Okay, yeah, that makes sense.
I think we have time for just one more question.
Nancy?
Yeah, okay.
Thanks a lot for that. So getting back to the question of prediction. So, a couple of years ago we introduced this concept of transcriptional risk scores, which is where you take an EQTL that overlaps with a GWAS, and then you sum the transcript abundance levels, Z-scores, and it does a lot better than genetic risk scores for prediction of disease progression. And so we've wanted to do that from a predictive basis, combining cis with trans, and the problem is that the body is responding. So it induces and suppresses a lot of genes once you get disease. And that's really interesting. You can use that, but I'm wondering if you've done anything in terms of adding the trans to the cis to get better predictors and what's needed for doing
So
it.
we're looking at that in primarily in blood, where there's enough data now that people are really getting good information on the trans predictions. And I think it's going to be a really interesting thing to try to work on. And I loved the idea of accumulating information on the levels in the way that you described. And I think for some kinds of phenotypes and diseases, that could work well. But I also agree, I mean, there's already a lot of really interesting information on the distinctions between the risk factors for development of disease and the risk factors for progression. And I think that's another important thing that we need to be working towards with all of this.
Thank you very much, Nancy. And now we're going to have Miriam Fornage speak to us about epigenetics.